Okay, I'm starting. Uh, Hi, I'm happy to be here today at the Cell Biology virtual event from LabRoots. My name is Ivan Robert Nabi. I'm a professor in the Life Sciences Institute at the University of British Columbia in Vancouver, Canada. I'll talk to you today about AI-based analysis of super-resolution microscopy, biological discovery in the absence of ground truth. The work I'll present is really a result of a crosstown interdisciplinary collaboration between my lab, located here at the University of British Columbia, and the computer science lab of Ghassan Hamarne, located at Simon Fraser University, also in Vancouver, but at the other end of town. And it all started with some really lengthy discussions between me, Ghassan, my student Ray Mang, and Ghassan's student Isma Kater in a coffee shop in downtown Vancouver. And this has led to what has proven to be a really long-term, I guess, for at least 10 years now, and team effort with lots of members of both labs being involved. I should say it's been really very interdisciplinary, a really strong learning experience, and also a lot of fun. So super resolution microscopy, I guess there's lots of different types of super resolution microscopy. There are three styles that are perhaps most prevalent and most available to most biologists. The first is single molecule localization microscopy, SMLM. You may have heard of this and going under names like STORM, PALM, or GSD. It can typically reach resolutions of about 10 to 20 nanometers in XY or laterally, and about 25 to 50 in Z. Now there's a new SMLM approach called MinFlux, which really has a quite incredible less than 5 nanometer isotropic resolution in both X, Y, and Z. The way single SMLM works is that you have isolated fluorophores. And so if you have an isolated fluorophore, you'll have an isolated point spread function, and you can predict the localization of the molecule based on the centroid of the point spread function. And what that means is that what we're generating is not really an image, but rather we're generating, when we apply SMLM, is really a point cloud, just a whole bunch of localizations in 3D space. And actually the output of a single molecule localization microscopy is actually an event list, an XYZ coordinates of different localizations. And you can see here, this is work from our lab on caveolin 1, and I'll talk a bit about this later, the incredible improvement in resolution that we see when we reconstruct an image in DStorm compared to the wide field image. Another approach is called stimulated emission depletion microscopy, or STED. STED essentially functions by aligning a laser in the shape of a donut that will deplete fluorophores, fluorescent emission, actually shifted to another wavelength so we don't see it, of the outside of the point spread function. So essentially all we're seeing, the effective PSF, is what's coming through the hole in the donut. And you can see down here in work from my lab on endoplasmic reticulum, the incredible improvement from confocal here in red to STED here in green. And STED can also be applied in 3D, where you align another donut in the Z position, and you can essentially improve by about twofold, maybe a little bit better than this, the numbers I'm showing here, where you can get a twofold improvement of resolution. Another approach that's really quite available commercially is structured illumination microscopy, where you have these grids which rotate and realign and generate moire patterns, which can be reconstructed to improve resolution, again about a twofold increase in resolution over standard confocal. So this is great. Super resolution microscopy still generates really nice images, but I would say what they're really doing is just taking the outer structure of a building and going with a little bit more detail and seeing higher detail of some features or some parts of the building so that you can get a better image. What we really want, and I would say we want as biologists, is we really want to know what's the structure, what's the inner structure, what's the design of the building. And the question is, how do we move past the image to get the inner structure or molecular architecture? And we would argue that the way to do that is using artificial intelligence. And so how do we apply AI to super resolution microscopy for biological discovery? And I say for biological discovery as opposed to using AI to better interpret the data we get from super resolution microscopy. And so classically and typically, AI is using what's called strong supervision. We're telling the computer, the AI, what, let's just say, oh, these are cat's ears, these are cat's eyes, these are dog's ears, these are dog's eyes. Okay, can you go find them? And AI is amazing. AI can find them. And if you annotate correctly and you give strong supervision, AI is quite incredible. The problem is that if we're doing new biology or we're doing research, we actually want to find things that we aren't known. And so we don't have means to really annotate, and we lack ground truth to train the AI. And so if we're using supervision, we have to have supervision approaches that don't depend on subcellular for the cell, subcellular object level ground truth, but rather we need alternative approaches. And the approach that we've applied is really weak supervision, where we don't tell the AI, hey, this is an eye or an ear, and find them. We say, oh, over there are the cats and over here are the dogs, and can you tell us what the difference is? And this approach has proven to be quite successful for us, at least in cell biology, since we have lots of groups and group labels that we can develop based on cell type and cell mutations or transfecting genes. And so we can generate lots of different group labels. And finally, I just say that we want an output that's more than classification. We don't want to know that they're dogs. We actually want to know why they're dogs. And this is something that's really important. And this is a challenge when we look at the different styles or approaches that we can use for AI. So classical machine learning would tell the AI, well, here we have a bunch of handcrafted features. Can you run this formula with all these features and characterize what you see in terms of these different groups? And this is what's called a short or a shallow recipe. And this generates, because we're giving it the features, it generates interpretable output that's suited for biological discovery. And I'll give you a couple of examples of that later on. Deep learning, which we've seen lots of, and we are using pretty much all of us every day these days, is really a deep recipe, and they have trillions of unknown operations and parameters, and it performs incredibly in prediction applications. It's really incredible how it works. And the challenge is that the decision processes are, to a certain extent, black box decision processes. There are lots of efforts and lots of improvements in developing explainable AI or XAI these days, and hopefully they will come. But still, when we apply it to biology, we're still at a point where there's still things to learn. And so we first of all, we published we've discussed this in a perspective that was recently published in Journal Cell Biology, and you're welcome to look at it. And we would argue that if we're applying weak supervision to super resolution microscopy, the first thing is to select trustworthy group labels that are grounded in the biology. They have to make sense to the biologist, so if we're going to apply them and apply weak supervision. We can then use the AI to identify different features across the groups, and that the AI will discover what's different. And then we have to go, we find that it's really important to go back and use a priori biological knowledge to corroborate whatever we discover or whatever the AI discovers. And so I'll talk to you today about three different biological examples that we've developed in which we've applied weak supervision. The first one is based on a software package called SuperResNet, which is applied to single molecule localization microscopy. And essentially what we did there is we really wanted to identify non-caveolar structures called scaffolds. And so we set up two groups where we compared cells, one which had caveoli, one which didn't. We then went and classified the structures that we saw. We validated it going back to the cryo-EM. And then we found, oh, well, hey, not only do we see caveoli, we also saw some new structures. And I'll talk a little bit about that more in depth later. We also developed another software package called MCS Detect, which we applied to 3D STED microscopy. Here we were interested in characterizing ER mitochondria contacts, or MERCs. and in particular characterizing contact sites which had these elongated contact sites that had ribosomes or ribomercs. We set up a software analysis that could do that. We validated it using known biology, not targeting regulators of these contact sites, and we discovered novel shapes of these ribomercs. And finally, I'll talk to you a little bit about our foray into deep learning, where we applied deep learning to viral infection, comparing the ER, the endoplasmic reticulum morphology in Zika virus transfected cells compared to mock transfected cells, and how we tried to identify the regions that were associated with the changes. The reality is that we still would like to know exactly, develop future approaches that are more explainable, that can really explain why these regions are chosen. And so the first story is SuperResNet, caveolin. Caveolin 1, Cav1, is a code protein for caveoli. These are 100 nanometer plasma membrane invaginations, and their formation requires this adapter protein called Cavin1, this little guy here in green. In the absence of Cavin1, Cav1 is localized to non-caveolar domains that are called scaffolds, and there's lots of evidence out there of functional roles for scaffolds, including in cancer progression. But the trouble is defining what these are has really proven difficult. By EM, you can do some labeling, and you can see caveoli labeling at the plasma membrane. But what are these? They look like just caveolin-positive flat membrane domains. By fluorescent microscopy, here in red, caveolin labeling gives lots of spots. So no, maybe the small ones are scaffolds and the big ones caveoli, but really it's really hard to tell. So to define the structure in caveoli and scaffolds by single molecule localization microscopy, in the absence of ground truth, we don't know what they are. We set up two group labels in which we compared PC3 prostate cancer cells. And these are cancer cells that have genetic ablation of Cavin1, so no Cavin1, no caveoli, and they only express, the Cav1 expression they have is located pretty much exclusively to scaffolds. Okay, and you can see here a really nice D-storm image of caveolin in these cells and the homogeneous distribution of caveolin to these spots in these cells. We compared them with species cells in which we had transfected Cavin1. So when we put back Cavin1, we now get caveoli, and so now we have larger clusters in these cells by D-storm, and now we have a means to do some weak supervision where we compare only scaffolds with cells that express caveoli and scaffolds. In the absence of ground truth, we use differential group labels, different cells, PC3 cells versus PC3 cells transfected with Cavin1, to validate machine learning identification from super resolution microscopy data sets. So what happened is when we applied SuperResNet and classification to PC3 cells, we essentially found two groups. And when we did the same thing, when we had PC3 cells transfected with Cavin1, PTRF is another name for Cavin1, we found four groups, and one of those groups, quite nicely, was the largest group, this blue group, and this blue group was corresponds to caveoli, consistent with the fact that these cells, these Cavin1 expressing PC3 cells, contain caveoli. And this is work from Ismail Kater, who started off way back when in that coffee shop I showed you earlier. We then went back and said, well, how do we compare what we have with what's known about caveolin-1? The recent, really quite elegant work has shown that caveolin-1 forms these really nice, stable disc-like structures containing the 11 Cav1s, and these correspond to what have been previously characterized as 8S disc-like complexes. And this 8S complex actually corresponds to one of the structures, or the scaffolds that we discovered by SuperResNet, which is S1A scaffolds, which have about 10 to 15 Cav1s per group. We also found that S1A scaffolds can come together to form dimers, S1B scaffolds. They can come together to form these hemispherical structures, which we call S2 scaffolds, and they can also come together to form these larger structures, the blue structures that I showed you before, that are more round and spherical and correspond to caveoli. And what was really nice is that when we took a look at our caveoli and compared them to what was known from cryo-EM about caveoli, there was quite a bit of similarity. By cryo-EM, caveoli are thought to be a dodecahedral complex composed of 12 caveolin 8s complexes, these 12 8s disks, and they're held together by a cavin net. What we found when we analyzed the modular structure of caveoli was that they were formed of 6 to 7 S1b scaffolds. S1b scaffolds, remember, are dimers of S1a scaffolds, so they're dimers of 8s complexes, suggesting that the caveoli we find have about 12 to 14 8s complexes or S1a scaffolds, suggesting a large degree of similarity between what we found using single molecule localization microscopy and supervised analysis and what was found by cryo-EM. And so what's the role of these scaffolds? And that's and I would say that's a good question. Early work from our lab showed that scaffolds really were an inhibitor, were the Cav1 domain, which inhibited EGFR signaling. And there's a lot of work been done on characterization of Cav1 regulation of signaling primarily via this caveolin scaffolding domain, a highly conserved domain. At the same time, another function of caveolin and caveoli was really as a membrane buffer. And this is work from our collaborator, Christophe Lamaze, where he showed that caveoli flattening when the plasma membrane is stretched protects the membrane from breakage. And we collaborated with Christophe to figure out, well, if when these caveoli flatten upon stretching, are they releasing scaffolds, and are these scaffolds functional? And so Christophe applied hypotonic shock to his cells, and he found that he saw flattening of caveoli. The caveoli he saw got larger from 80 nanometers to about 120 nanometers. He also saw, using supervised analysis, fewer caveoli in the hypotonic shock treated cells, and an increased abundance of scaffolds, all three types, S2, S1b, and S1a. He validated this result with Giannoni and Borsier in Bordeaux, who applied single particle tracking to Cav1, and what they found is upon hypotonic shock, they saw that the Cav1 in the membrane was far more mobile, consistent with the fact that caveoli are known to be highly immobile and releasing scaffolds, and therefore upon hypotonic shock, they release scaffolds, which become far more mobile. And so, and when Christophe went to look and see whether or not the Cav1 is interacting with JAK kinase, a receptor known to be regulated by Cav1, he found that upon hypotonic shock, there was increased association of the smaller scaffolds, S1a and S1b, with JAK kinase, and he showed that this is inhibiting JAK kinase activity. And so the idea is that we have a steady state, we have caveoli, we have maybe fewer scaffolds, we have an active signaling effector, in this case JAK, but when we get membrane stretching, caveoli flatten, they release scaffolds, and they interact with the signaling effector, and they inhibit its activity, suggesting that this is a mechanosensing signaling paradigm where the cell senses stretch, transmit that stretch by flattening of a cell surface structure, releasing these heterotypic complexes between signaling effectors and caveolin scaffolds, and thereby impacting the signaling response of the cell. Okay, so now I'm going to go to part 2. I'm going to talk to you about MCS detect and ER mitochondria contacts. Mitochondria ER contacts, MERCs, were originally identified biochemically as membrane-associated MAMs. You can see them here, and they're one class of contacts that the ER forms with many organelles. However, really identifying what these MERCs are by fluorescent microscopy is challenging. EM is great, but fluorescent microscopy can be difficult, particularly because the distance between these two organelles, about 10 to 60, maybe 80 nanometers, is far below the resolution of optical microscopy and even below the resolution of 3D super resolution microscopy. If we want to segment, and we're segmenting two channels, ER and mitochondria, they're very subjective and difficult, and the ER is particularly difficult to segment by fluorescent microscopy. And finally, there are biofluorescent complementation systems which use linkers of various lengths to bring the organelles together or monitor and detect these organelles, but they may promote, they may stabilize, and to what extent the contacts and to what extent they are valid reporters is really challenging. And so we set up a system. This is Ben Carduin, another student in Ghassan's lab, and he set up a system whereby he essentially used a Laplacian operator to detect local signaling differentials and scan this Laplacian operator across throughout the 3D volume of the image stack. And these were, we then applied a negative Spearman correlation between two different channels to really detect where the relative labeling intensity of the two channels changed in tandem. And so where they change in tandem is essentially a site where these two organelles come into contact. And what you can see here in this simulation is that as the red and the green balls come together closer and closer, the Spearman correlation of the Laplacians increases. And this is the basic principle underlying MCS detect, which is really a segmentation-free approach to detect organelle contact sites from 3D super resolution image stacks. And so we had to go back to biology, set up a test case where we could test whether or not the biology, whether or not the system actually generates valid and correct data. The system we used was these HT1080 cells, which we had previously shown expressed these really elongated and elaborate mercs, which are these, we have these ribosomes on them and that we call ribomercs. And we had previously shown that if we knock down the GP78 ubiquitin ligase, that we lose or reduce the extent of these ribomercs. They were also shown to be very abundant in liver, and you can see these beautiful liver where they wrap around the liver, and they were called RAP-ER. When we did some EM of COS7 cells, we found that the COS7 cells had no ribomercs, and that pretty much all their mercs were smooth. And so we set up a test case for weak supervision, whereby we compared differential group labels of HT1080, abundant ribomercs, compared to COS7, few ribomercs. When we applied MCS detect, and we did 3D STED, a deconvolution, and processed the images by MCS detect, we could get here, you can see there's a large increase in the extent of contact sites in white from COS7 to HT1080. Mitochondria are in red, endoplasmic reticulum in green. We saw a similar increase in contact sites if we transfected COS7 cells with an outer mitochondrial membrane ER linker. And you can see here the increased extent of contact sites on the mitochondria compared to COS7 cells, untransfected COS7 cells. We then tested whether or not GP78 could regulate ribomercs. And you can see here, if we knock out GP78 and HT1080 cells, we see reduced extent of contact sites. If we overexpress GP78, we induce contact sites. We then went to the biology again, and we tested whether or not known regulators of ribomercs are impacting our detection of contact sites. Mitochondrial SYNJ2BP and ER-associated RABP1 are known to be ribomerc tethers, published from the Ting Lab in 2017. We first went to EM. We knocked down RABP1, one half of the tether, and we saw a dramatic reduction in ribomercs in both HT1080 cells and GP78 knockout cells, consistent with what was previously published. And when we went to MCS detect, we saw essentially exactly the same thing, a dramatic reduction in contacts in both HT1080 and GP78 knockout HT1080 cells. So what we argue is that this is dual control of ribomercs. On the one hand, we have a tether that's required, RBP1 interacting with SYNJ2BP, required to form the ribomerc. At the same time, it suggests that GP78 activity and ubiquitin ligase activity is regulating the size of the ribomerc. And we then went to see, well, what do these ribomercs look like? What can we learn about their shape? And so we went and we chose to look just at the top 5% in size and volume of ribomercs. And you can see here, let's get the movies going. And you can see here that in HT1080 cells, you see these really elongated tubular shapes. These are recapitulated upon overexpression of GP78 in COS7 cells. And you can see here these very nice elongated tubular shapes that really correspond to the tubular matrix that was previously shown to be these tubular interacting tubules from the Lippincott-Schwarz lab by Fib SEM. What was interesting is that when we took a look at the cells expressing this linker, the contact sites were flatter smaller, and different in shape, suggesting that different contact sites can form, and that these tubular ribomers have a distinct tubular morphology. This was published in JCB, and we have the cover, and you're welcome to look and take a look at this paper, and certainly we're happy to respond to any comments. Finally, I'm going to go and talk a little bit about deep learning and our application of deep learning to virus-infected cells. This was done a few years ago before the COVID-19 pandemic, and so we were focusing on Zika virus. And I'll just talk to you a little bit about how we use deep learning and host cell ER organization to identify Zika virus-infected cells. Transfection, this is an ER, we're using an ER-MOX GFP reporter to label the ER. And as you can see here, when we infect a cell, show this sort of a concentration, crescent-like concentration of the ER around the nucleus, where we find lots of double-stranded RNA, that's viral double-stranded RNA, and that is indicative of viral replication sites. We set up a convolutional neural network, a CNN, in which we took 28 cells of mock infected and Zika virus infected, and we processed them via training sets and validation sets, and to be able to identify if any particular STED frame, STED image, looking at each frame, could be predicted to be either infected or non-infected. The deep learning, even from only about 28 cells, did a pretty good job, in which we got 80% correct identification of infected versus mock frames. And then we thought, okay, this is great. We have another case where we have weak supervised learning. Can we now go and learn what the deep learning is doing? And so we use class activation maps, or CAMs, to highlight the regions that are identified as discriminatory by the deep learning. And the red ones are the ones that correspond to how the cells are identified as infected. The green guys, how they're identified as non-infected. And you can see that for the Zika virus infected cells, it's really focusing in on that dense region. So to see is focusing on this peripheral network region, which is pretty good, because that's what we saw before to be altered. And when we actually went into to focus on the region, we found these really dense tubular networks, which co-localized with the double-stranded RNA from the virus, and also showed the same tubular matrix or organization that I showed you previously for the ribomers. We also went by EM, and you can clearly see that the ER organization changes. We have nice parallel sheet-like structures in the rough ER, while in the Zika virus, the mock infected cell, or the Zika virus infected cell, we see all these convoluted tubules. And so that's great. And so the deep learning has identified regions corresponding to what we saw, and they're able to really discriminate the virally infected cells, but they don't really explain why the region is selected. And we have to interpret. The biologist really has to interpret and go back and say, oh yeah, these are tubules and these are tubular matrix, and so it's not really telling us what's going on. It doesn't provide an interpretable output, and I think this study was done a few years ago, and I think there's been lots of advances in explainable AI. XAI, and I think we're really looking forward to developing new approaches whereby the deep learning, which is incredibly powerful deep learning, can provide interpretable approaches that we can understand, and we can learn new biology from deep learning. And so, in summary, I think I've tried to present you a number of examples about how we can define the molecular architecture or the inner structure of the cell to develop new biology, new biological discovery in the absence of ground truth. I think the basic principle that I presented is the application of weak supervision to group labels that are defined by biology. And in our case, we're using different cell types: transfected cells, cells that express one feature or another, infected cells. But this can apply to pretty much any defined group label that for any selected biology. We do a comparative analysis of groups, and we can use machine learning and deep learning to identify features that are typical of one group or the other. I've also presented two software packages that we've developed. One is SupervisedNet for network analysis of SMLM point cloud data, and I think this is a really powerful approach to obtain mesoscale Mesoscale, I don't know, 10 to, between about 100 nanometers or so, less than 100 nanometer size, not atomic scale, but mesoscale structural biology analysis. So we really think it's a powerful tool to look at how protein oligomers come together to form structures within the cell. I also presented MCS Detect, sub-pixel resolution analysis of voxel-based super resolution data. And that represents, I think, a novel approach and a new approach, a non-invasive approach to identify interaction between subcellular organelles. Future challenges for the application of AI to super resolution microscopy, and I say here for biological discovery, are to get weakly supervised AI to not only tell us why groups are different, but to specifically identify discriminatory structures, to say, hey, these structures are really different, and this is why we're identifying these groups as being different. And I think we really want to extend this to deep learning and determine the biological basis for deep learning discrimination between groups, to go beyond PAMs and use and implement new approaches and explainable AI that are being developed. And finally, we want to make the software accessible to biologists. You can find the software or the current versions of this software and other softwares, and you're welcome to download it and contact us for support at this GitHub site, Nanoscopy AI. Finally, I'd like to thank all the members of my lab and also Ghassan's lab, who've done, and I mentioned a lot of them, and who are really instrumental, as well as collaborations at UBC with François Jean, and also with Christophe at Curie, and also now with Ismail Kater. And finally, I would like to say thanks to the funding support, thanks to LSI Imaging, the imaging facility in the Life Sciences Institute where we do our work, as well as to Moshe Safdie for the drawings of the marine Bay Sands Art Science Library. So thank you very much.